Imyoo. We're a public benefit corporation spun out of here from Caltech. And my journey into the space, actually, I never thought I'd end up here. I was in electrical engineering, doing space stuff. I worked at NASA JPL, and then I got diagnosed with an autoimmune condition. And that whole pathway of getting diagnosed, getting treated, was really eye-opening. How are we supposed to go to space if this is the level of healthcare that we have now? So I decided to, after working at NASA for a while, spending nights and weekends at Caltech, to go into grad school, and this is really where this idea of Imyoo came about. So I'm going to talk about how do we enable longevity through direct-to-patient molecular immune tracking. And of course, longevity is a very personal thing. It means a different thing to everybody. I'd be curious to hear yours if I see you around. For me, it's living long enough to go to space. And I think longevity is specifically interesting in case of space because that gets accelerated when we're up there. So it's one of the other reasons I'm actually interested in longevity. So what we're building is we are working directly with, we call them patient scientists, to get their molecular stories. So we use this small device where patients self-collect capillary blood whenever they're feeling their symptoms, and then we're generating really high resolution data from that. So what it means to be a patient scientist is to be involved in research for getting therapeutics and tests developed. Not just, hey, I'm going to take some blood samples from you, and then maybe 10 years later you will see something. No, you should be there at every step. You should be notified what happens to your data, why it happens, and you should have autonomy in that. So we built these kits where we're looking at many molecular signals from a few drops of blood. It's not Theranos. This is all research right now. And I would say a few drops of blood. So this is a self-collection kit that we send to patients. If you're somebody who's doing intervention or are interested in getting more molecular data for your studies, whenever you're introducing intervention, come speak with me. So patients self-collect blood, they send this kit back to us, and we return some research data back as well as analyze molecular data. So some of the things that we were able to figure out is how do we do these research-grade assays on small volumes of blood. So when we've been able to do single-cell RNA sequencing, genetics, immune repertoire sequencing, which tells you what kind of things you're predisposed to, and cytokines, which kind of tell you, it's just how your immune system talks to different organs in your body. And using this system, we've been able to build a molecular atlas across many different people, diseases, and interventions. We have now over 600 people, profiled over 5 million cells, and have over 2,100 samples biobanked. And we collect really rich metadata about lifestyle, therapies, comorbidities, but also these patients talk to us. They tell us whenever things happen to them naturally in their life, not just a rigid kind of forms that you fill out at a doctor's office. So we've been particularly involved in autoimmune diseases like IBD, RA, psoriasis, lupus upcoming, and interventional data sets. And so we believe that by scaling this approach and this data, we'll be able to answer questions like, who's my molecular twin? What worked for them and what didn't work for them? When is my immune system vulnerable and what can I do to strengthen it? And did these interventions really work for me? What happened to my immune system? Does it remember this intervention? Okay, that's cool, but why is direct-to-patient molecular immune profiling important for longevity? So kind of going back to definitions for me, on a more kind of clinical analysis, longevity means having the power to intervene in your biology to control your health span. And in clinical care, it means using a validated set of tools, biomarkers, protocols, and interventions to achieve your desired health span goals. Okay, let's break it down. Why direct-to-patient? So direct-to-patient is important because traditional studies are very rigid. If you're having a flare, if you're having an adverse effect, or maybe when you're feeling amazing, you can't just contact your site, your doctor, and say, I want to donate blood. But we really want to enable people to be able to capture these really, really precious moments that don't usually get captured by clinical studies or whenever you go to a doctor. So we really are empowering patients to collect whenever they're experiencing these things, so we can develop more personalized and more precise therapies. Here you see an example of somebody driving their kit over to FedEx on a bike, and we have a network of patients that we work with directly. Another one is why molecular. Routine blood tests are nice, but they're you can't even see here, it's a tiny little red dot there, but there's millions of unique molecules that we don't utilize. So there's an ocean of actionable biomarkers that we haven't tapped into yet, and we really believe by combining this routine approach with these research-grade assays, we can get to something very meaningful. And then why is immune profiling? So your immune system is your systematic signature of what's going on with you. So your environmental response, aging, resilience. So here you see an example. This is actually me, I'll say. I have this myomycin 2 gene in my natural killer cells that look very different from everyone else. And this is actually a longitudinal study, AM to PM, for 3 to 4 days that we've done. And I'm consistently different from everyone else. So these are the kind of things that we should be looking at when thinking about developing new therapies and tests. And what's really cool is that this is also accessible by blood. Okay, go back. Another thing that was mentioned earlier in a panel done by Tom is there's also incentives, and this is something we don't appreciate. We have to incentivize people to participate and give their data. I think 23andMe kind of took advantage of that. So what we're doing that's unique here before we commercialize any tests or therapies is anybody who donates blood to our studies gets their research data back, but we also believe should get their revenue back. So if, for example, you have participated in our study, say you did an intervention, we generate this really rich data set, and if we share that data or samples with a therapeutic company or a biotech, you should get some of that revenue sharing back. So that's one other unique thing that we're innovating on as we're building our products in this space. I want to show you some examples of ways that people and we have used our approach. So this is number one. This is our autoimmune flare study across inflammatory bowel disease and rheumatoid arthritis patients. We went directly to patients, so 65 patients with these diseases across 6 time points. So 3 time points during a flare and 3 time points during a baseline. Another cool thing with this approach is we were actually able to match demographics of the US by going direct to patients. So we're kind of, and this is one issue that happens with longevity, is like, how do we go beyond the bubble? How do we make it more accessible? Come to people where they are. So we were able to collect this really rich data set, single-cell RNA sequencing, immune repertoire, cytokine, their infusion schedules. And we noticed a very interesting thing. This is very preliminary. We noticed there was 5 patients on a drug called Imyoo that prevents pathogenic T cells from being trafficked into a blood. However, these 5 patients are still flaring. So the therapy didn't work every time. So you had a situation where they had an infusion and they were fine 4 weeks after, but sometimes they had an infusion and 4 weeks after they ended up in a flare. So we're like, what is different between these 2 different situations? And we noticed that one of the subunits that makes up the Imyoo's target is actually downregulated specifically in CD4 T cells in people who are not achieving remission. And so you can see here these 4 patients that these T cells, the IgGB7 gene is downregulated. And my hypothesis is that the drug is not preventing these pathogenic T cells from being trafficked into the gut tissue. And this is something that you would totally miss if you did a cross-sectional study. For example, this person's baseline looks like this person's flare. So that kind of, highlighting what Nathan was saying is like longitudinal is really important for these types of things. Another thing we're able to see is that flares are very different between the baseline samples as well. So whenever you are sick, you look different. You're not just all sick the same way. So it's really important to kind of look, what is the difference? And also really use this really rich data set to define molecularly the disease instead of just symptoms. So here's another use case I want to share with you guys. So this is a group of, this goes more into kind of territory of citizen scientists. You're maybe familiar with long COVID. There's another disease called chronic fatigue syndrome where they experience very, very similar symptoms where they're bedridden, can't get out of bed, very sick, still not understood what exactly that disease is. And they reached out to us. It was a group of 2 patients, and they have a community of 500 or more. And they actually did a longitudinal study where they tried antibiotic, which helped them recover a little bit before. So we actually collected many, I think this was 7 or 8 time samples. So before they tried the intervention, during and after. And what was interesting is that, specifically in CD14 monocytes, we were able to see CCL4 expression being downregulated, and it continued to, and that maintained because these patients reported feeling better after doing this self-intervention. So, and this is where we all biohack, but we need better biomarkers to tell us, is it actually working? So for them, this was kind of a promising hope of that. Okay, so just to kind of wrap things up, I believe that one day the world will use molecular data such as this to personalize health day to day, and we are really building the data engine and the infrastructure to enable that. So where we are is Imyoo, we are building an infrastructure to collect high-resolution molecular data directly from patients across the US. Hope to be expanding internationally soon as well. And we are currently, we're launching these studies all the time, so if you're doing intervention, please come see me. And we're going to be launching at-home tests. We're specifically focusing on autoimmune diseases right now, but also happy to work with other folks in the aging space as well. I do want to leave you guys with a few things. What are some challenges to bringing new tools and things like this to market? There's incentives. What is in it for the patients to contribute their samples, their time and data? There's actionability. Having an actionable biomarker is powerful, but having loads of data can be anxiety. So it's really important to actually, take these biomarkers that you find and validate them and make them useful for patients. Don't just throw this data at them. And also adaptation. At-home testing is not widespread. There needs to be a call for action for healthcare to actually adopt these things as well. Okay, I will say thank you to my team. One thing I want to point out is that we actually have patients on our board that contribute to these study designs and making sure that we do it in an ethical way. Yeah, thank you. And any questions? So we spread out costs in different ways. So we can do biobanking, that's a separate cost, and then we can run single-cell array sequencing, immune repertoire, whatever you choose. The biobanking part is a few $100s, and then the rest depends on kind of what you choose, how many cells, how many genes, and say if you do this with your patients, they would get some of that revenue back as well. Okay, come find me after, yeah.
I saw, sorry I missed this, but yeah. There's is a program for the basal levels, basal health available?
Baseline.
Yeah.
Yeah, so anybody can enroll in our baseline study. It's free. We're not charging you. We will give you your report back. So we're ideally looking for people who are trying interventions. So if you're starting a new diet or new peptide or whatever, we prioritize that, but anybody can enroll.
Great. Even if you move to a different location,
yes,
environment effects.
That's the beauty of it. Actually, in our study, patients actually changed locations, and that didn't matter for us, right? Because we could just ship directly to them. Yeah.